Buenos días a todos los presentes. A nombre de la Facultad de Ciencias Biológicas de la Universidad Nacional de Trujillo, les damos la cordial bienvenida a la apertura de este evento académico organizado por los docentes de la Cátedra de Biología Celular y Molecular y la promoción 61 de la Escuela Profesional de Ciencias Biológicas. En esta mañana nos honran con su presencia autoridades de nuestra alma mater, quienes nos acompañarán durante la inauguración de la quinta edición del Simposio Internacional de Biología Celular y Molecular. Saludamos y agradecemos la presencia de las autoridades de la Facultad de Ciencias Biológicas de la Universidad Nacional de Trujillo, Perú. Doctor Eber Mas Robles Castillo, decano de la Facultad de Ciencias Biológicas de la Universidad Nacional de Trujillo, Perú. Doctor Peláez, director del Departamento Académico de Ciencias Biológicas de la Universidad Nacional de Trujillo. Asimismo, saludamos la presencia de los docentes de la Cátedra de Biología Celular y Molecular de la Facultad de Ciencias Biológicas de la Universidad Nacional de Trujillo, Perú. Doctora Fátima Zavala de la Cruz, coordinadora de la Cátedra de Biología Celular y Molecular y presidenta de la comisión organizadora de este simposio. Doctor William Blas Cerdán, miembro de la comisión organizadora de este simposio. Doctor César Narcés Díaz Sánchez, miembro de la comisión organizadora de este simposio. Doctora María Noemi Llanos Román, miembro de la comisión organizadora de este simposio. También saludamos a los alumnos de la promoción 61 de la Escuela Profesional de Ciencias Biológicas, Facultad de Ciencias Biológicas de la Universidad Nacional de Trujillo, Perú, miembros organizadores de este simposio. Al público asistente en la sala, les recordamos mantener sus micrófonos y cámaras apagadas, con el fin de evitar interrupciones durante el desarrollo de las actividades. Al público que nos sigue en la transmisión en vivo por nuestra página oficial de YouTube e Instagram, les agradecemos por su atención y acogida. A continuación, invitamos al doctor Freddy Peláez, director del Departamento Académico de Ciencias Biológicas de la Universidad Nacional de Trujillo, Perú, para dar inicio a las palabras de bienvenida.
Bien, muchas gracias. Buenos días con los presentes. Bueno, primero agradecer a la comisión organizadora de este quinto Simposio Internacional de Biología Celular y Molecular, que está liderado por la Cátedra de Biología Celular y Molecular, integrado por los doctores, la doctora Fátima Zavala de la Cruz, William Blas Cerdán y el magíster César Díaz Sánchez, ¿no? Y también a los estudiantes del sexto ciclo, específicamente del curso de Biología Celular y Molecular, de la Escuela de Ciencias Biológicas, promoción 61, que tienen a cargo toda esta organización, que en esta vez, como ya se mencionó, es la quinta versión, ¿no? La quinta edición de este simposio. También un saludo a todos los docentes presentes, docentes he visto de la facultad, del Departamento de Ciencias Biológicas, del Departamento de Química Biológica y demás integrantes y participantes en este simposio. Bueno, para nadie es un secreto ahora que la biología molecular pues está en el punto más alto tal vez, ¿no? Y sobre todo después de la nefasta fase que pasamos por la pandemia, donde vimos esa carrera acelerada que se generó para conseguir las vacunas, y indudablemente ahí es donde apareció en forma oportuna tal vez estos mecanismos que hoy se han visto sus logros en la obtención muy rápida de estos elementos que nos salvaron la vida ya de muchísimos millones, a pesar de que se habían perdido ya millones también de vidas, ¿no? Pero eso es lo que está ahora trabajando la biología molecular. Y la biología molecular como rama pues de la biología no solo trata de ver la estructura molecular, diríamos así, la estructura morfológica que pueda tener la composición, sino más que todo las funciones, ¿no? Y cómo es que esas tienen repercusiones, esas macromoléculas que se forman tienen repercusiones en la fisiología de todos los seres vivos. Y hoy es donde, bueno, los estudiantes de sexto ciclo pues van a ampliar esa visión gracias a la participación de excelentes expositores de muchos países, ¿no? Me olvidé tal vez en el saludo cuando dije buenos días, pero no me olvidé indicar que para otros lares, de repente, porque tenemos conectados del Japón, de Italia, de Suiza, me parece, entonces que ahí ya no es buenos días, estaremos en buenas noches o buenas madrugadas, no sé qué podríamos decirlo, no he revisado el calendario, pero sí es el nivel que tienen los ponentes, ¿no? En los cuales hay que destacar para el Perú que hay dos ponentes nacionales, bueno, que trabajan y que están estudiando en el extranjero, y entre ellas una investigadora nacida en la Facultad de Ciencias Biológicas, en la Escuela de Ciencias Biológicas, que es la doctora Irene Julca. Ya tendremos la oportunidad de escucharlos, ¿no? Entonces, a nombre del Departamento de Ciencias Biológicas, a la cual pertenece la Cátedra de Biología Celular y Molecular, les damos la bienvenida y que lo que se hayan planteado, los objetivos que estén tanto para los organizadores como para los demás participantes, pues sean de lo mejor y cumplan con todas esas expectativas que se han forjado. Muchísimas gracias otra vez y bienvenidos. Y doy por aperturado este simposio de biología molecular y celular y molecular. Muchas gracias.
Agradecemos la participación del doctor Peláez. Para dar inicio con nuestro acto protocolar, invitamos al señor decano de la Facultad de Ciencias Biológicas de la Universidad Nacional de Trujillo, Perú, el doctor Eber Max Robles Castillo, para brindar las palabras de apertura. Adelante, señor decano, lo escuchamos.
Buenos días con todos, ¿me escuchan? Hola. Sí, ¿no? Muy
Sí.
bien. Los gestos que me... Muy bien. Un honor estar aquí con todos ustedes. En primer lugar, con el doctor Freddy Peláez, director del Departamento de Ciencias Biológicas de la Facultad de Ciencias Biológicas de la Universidad Nacional de Trujillo, Perú. Igualmente está aquí con nosotros la Cátedra de Biología Molecular y Celular y Molecular de nuestra facultad, liderada por la doctora Fátima de la Cruz Zavala y todo su equipo de docentes. Igualmente, la presencia de nuestros estudiantes de la promoción que está organizando este quinto Simposio Internacional de Biología Celular y Molecular, un evento organizado por la Cátedra de Biología Molecular y Celular del Departamento de Ciencias Biológicas de nuestra facultad de la Universidad Nacional de Trujillo. De verdad, yo estoy muy contento y orgulloso de la realización de este evento, porque no es sino una muestra más de querer superarse y querer salir adelante en esta área tan importante de la ciencia que nos permite día a día estar viendo los elementos moleculares y de parte de los aspectos celulares de la biología, ¿no? Muy íntimos, que incluso han sido motivo de algunos premios Nobel a nivel mundial, ¿no? Y esta área es muy importante, muy emergente y siempre está en los últimos avances de lo que se refiere a lo que es la parte molecular de la biología celular, ¿no? Y de verdad que nuestra facultad se pone muy contenta porque estos eventos que involucran no solamente la parte nacional, sino también internacional, a grandes personajes como ya lo ha mencionado el doctor Peláez, ¿no? De Japón, de Estados Unidos y de otros lugares, ¿no? Y sobre todo que algunos de ellos son estudiantes egresados de nuestra facultad, de nuestra escuela, y que están haciendo, digamos, ciencia en esa área tan importante de la biología. Yo, de verdad, mi granito de arena aquí es apoyar para que estos eventos continúe, digamos, en su realización, ¿no? Y de verdad que agradezco al equipo organizador, igualmente a los ponentes internacionales, nacionales, ¿no? Y a todos los que aquí se han dado cita en este evento, porque no hacen sino más bien resaltar que esta organización tiene mucha importancia, ¿no? Para nosotros es muy relevante porque eso significa una tarea dentro de los eventos científicos y por supuesto de acreditación de, digamos, nuestra escuela. Por eso estamos siempre apostando a que se realicen eventos científicos de esta naturaleza. Igualmente, importante conservar la calidad de los ponentes y por eso también nuevamente punto a favor para la acreditación de nuestra carrera y la vigencia sobre todo, ¿no? Ya tenemos 62 años en Trujillo, digamos, como carrera existente de biología, ¿no? Y entonces, a seguir adelante. Y yo la verdad que estoy, como repito, vuelvo a repetir, muy contento y agradecido por este simposio, quinto simposio internacional de biología celular y molecular. Con las felicitaciones del caso, yo creo que les damos pase a que continúen en esta actividad tan importante. Adelante, muchas gracias. Buenos días.
Agradecemos la participación del doctor Robles. Agradecemos la participación de las autoridades que nos acompañan en esta mañana, así como a los investigadores que nos honrarán con su participación como ponentes, y a todo el público asistente que participará durante las diversas ponencias presentadas en este simposio. Al público asistente en sala y al público que nos sigue en la transmisión en vivo por nuestra página oficial de YouTube e Instagram, les recordamos que el programa de este evento está publicado en la página oficial de Facebook. Además, les informamos que este evento es gratuito y de libre acceso y con emisión de certificado que evidenciará su participación para ser merecedores del certificado, en mención que se tendrá que evidenciar con participación como asistentes, llenando al menos 7 de 9 formularios que corresponden a cada una de estas ponencias. Los enlaces de los formularios se les estará compartiendo en un oportuno momento en cada una de las ponencias.
Good morning. Following with the program, it's a pleasure to present Dr. Aiko Sada, who is professor at the Medical Institute of Bioregulation at Kyushu University and in the International Research Center for Medical Science at Kumamoto University of Japan. Aiko Sada obtained her PhD in 2011 from the Graduate University for Advanced Studies in Japan. After graduation, she joined Dr. Tudorita Tumba's lab at Cordial University as a postdoctoral fellow, where she worked on skin stem cells. When she returned to Japan in 2016, she continued studying skin stem cells dynamics in tissue, homeostasis, regeneration, and aging. At present, using mouse genetics and 3D culture system, She is investigating the regulatory mechanisms of skin stem cells under physiological or pathophysiological conditions. Dr. Aiko Sada will give the talk titled Reliving the Stem Cells Dynamics in Skin Regeneration and Aging. Dr. Aiko Sada, welcome to this event. We appreciate your participation and your support. Please keep in mind that your talk should last at most 45 minutes, so then we could proceed to answer the questions that the audience may have for around 10 minutes. We would like to remind the audience present in this room to keep their microphones and cameras turned off to avoid interruptions during the presentation. Please submit your questions via chat during the session, so they can be addressed at the end of the talk. Additionally, a link of the attendance form will be shared during the presentation for you to register your participation. Al público presente en sala le recomendamos mantener sus micrófonos y cámaras apagados para evitar interrupciones durante la exposición. Si tuvieran preguntas, estas deberán ser formuladas mediante el chat durante el transcurso de la presentación y serán absueltas al finalizar la ponencia. Además, les comunicamos que durante el transcurso de la exposición se les estará compartiendo el enlace del formulario para que registren su asistencia. Dr. Aiko Sada, please, you can start with your presentation. Thank you very much.
Thank you very much for your introduction. Let me share my screen. Sorry. Oh, it says I'm not allowed to share my screen somehow. Could you help me sharing my screen? I think I'm not have access to share my screen.
Good morning, Dr. Aiko Sada.
Hi,
Yes.
hi, good morning.
Just good morning. Just let us a few seconds to check what's happening. Thank you very much.
Sorry. Yeah, thank
No
you.
problem. I guess it's something related with the authorization. Just wait a few minutes. Thank you. I'm sorry for the inconvenience.
No, sure. Thank you.
So now you can try. You can go to the...
Sure. I will try.
Okay.
Sure.
Okay, so now we see your screen, your whole
Great.
screen.
Great.
So you can.
Okay.
Thank you. Thank you.
Okay, great. Thank you very much. Can you hear me well?
Yes, we do.
Okay, great. Thank you very much for giving me an opportunity to present my work today. My name is Aiko Sada. I'm from Kyushu University Medical Institute of Bioregulation. So my university is located in southwest part of Japan, the city of Fukuoka. I'm very happy to be here today to share our work. So my laboratory has been focusing on stem cell dynamics in skin regeneration and aging. We are particularly interested in how heterogeneous stem cell population play a role in physiological conditions as well as pathological conditions such as skin aging and diseases. So from stem cell biology point of view, our lab have been focusing on the slow cycling nature of tissue stem cell. So generally, tissue stem cell have been considered to be a slow cycling in nature. So this unique property of tissue stem cell allows them to divide slowly to protect them from replication-induced DNA damage, tumorigenesis, and aging. And this slow cycling stem cell found in many different mammalian tissue, including skin and the hematopoietic stem cell system and other mammalian system. For example, in the skin, slow cycling stem cells are located in the bulge region, and they act as a hair follicle stem cell and contribute to cyclic regeneration of hair. Similarly, ocular surface epithelium of the eye, the slow cycling stem cell are located in the limbus region, the boundary between cornea and conjunctiva, and those cells are also known to act as stem cells. So this is generally considered to be a mechanism to protect tissue stem cell and accumulate from DNA damage, accumulation of mutation, and telomere shortening. But there are other models suggest different role of slow cycling cell. For example, slow cycling cell could be a reserved population that do not actively participate in tissue homeostasis, but they are activated upon injury. The other model suggests that those slow cycling nature are the stochastic thing. So some cells divide more or divide less, but in terms of stem cell potentials, they may be equal in their property. So we know that the mammalian tissue have slow cycling cell, but we still don't know what is the biological significance of having slow cycling cell in the tissue, then how they behave, and then how they are regulated in different contexts. So in my team, we use skin as a model system. Skin consists of the uppermost epidermis and dermis. Epidermal compartment accommodate different populations of stem cell. In the hair follicle, there is a hair follicle stem cell located in the bulge region, and they contribute to cyclic regeneration of hair. In contrast, in the interfollicular epidermis, epidermal stem cell reside in the basal layer, and these cells self-renew and differentiate upwards to form spinous, granular, and conified layer and contribute to barrier function of skin. So skin stem cell research has advanced greatly in past 20 years by mainly using bulge hair follicle stem cell as a model system. However, in the interfollicular epidermis, it remains poorly understood which cell act as stem cell in the epidermis, and then whether slow cycling cells are the tissue stem cell. So in our study, we try to address this question by using a transgenic mouse system, H2B-GFP. So here, in this mouse model, we can visualize cells with different cell division frequency using the GFP reporter. So here, we genetically label epidermal cell with H2B-GFP reporter, so cells become green, so in epidermis specific manner. Then since this is a TETOP system, so when mouse are treated with doxycycline, it stops the transcription of H2B-GFP. Then when cells divide, cells dilute GFP signal after each division. So first dividing cell will eventually lose GFP signal, whereas slow cycling cell will retain GFP signal for a longer period and detected as level retaining cell, or LRC. So by using this mouse system, we visualize the slow and fast dividing cell in the mouse tissue. So here we show mouse epidermis, skin epidermis, by looking by a hormone staining. So here we separated epidermis as an intact sheet and stain as a whole and observe by confocal microscope. So when we, usually observe skin tissue by section view, but here we look at the epidermal sheet as an intact sheet and then looking from the basal side like this. So in this picture, so without doxycycline treatment, you can see all the epidermal cells are labeled in green. However, after 6 weeks of doxycycline treatment, now we can appreciate this very beautiful pattern of GFP. So here, the green cells are the level retaining cells, so they are dividing slowly. And blue cells, those are the cells that have divided GFP signal, so those are rapidly dividing cells. And those, so in this picture, so each small dot represents one cell. So meaning that this is a cluster of 100 cells, making a compartmentalized pattern in the tissue. So this is very interesting. So this observation suggests that in the mouse skin epidermis, the slow and fast cycling population are highly compartmentalized and make this very beautiful pattern. Then we are very interested in the phenomenon, and we first isolate those GFP positive and GFP negative cells, and then characterize their molecular identity. And then next we address whether the slow cycling cell and fast cycling cell act as stem cell or not. So to address this question, we perform lineage tracing using the newly identified stem cell marker. So in this experiment, we genetically label cells of interest by using a gene of interest identified by RNA-seq analysis. So here we injected tamoxifen to mice and activate Cure to label cells genetically and then track their fate. So once Cure is activated, tomato reporter is turned on, so we can label the cells of interest by using the promoter of gene. So here, if the label cell acts as stem cell, those cells self-renew and differentiate upwards to form epidermal cell. Then since these cells have self-renewability, so this epidermal clone will maintain long-term in the tissue. In contrast, if the label cell acts as short-lived progenitor cell, these cells divide rapidly, differentiate, but since they do not have self-renewing ability, those epidermal clones are eventually lost from tissue. So in this way, we can distinguish stem cell and non-stem cell in the intact tissue. Then by using this system, we address whether the slow cycling cell and fast cycling cell act as stem cell. So here we identify DLX1. So this is a transcription factor identified by RNA sequencing, and then used as a slow cycling cell marker. Then SLC1 and 3, this is a glutamate transporter and used as a fast cycling cell. Then by injecting tamoxifen to mice, we successfully label DX1 and SLC1 and 3 positive cell in the mouse skin tissue. And then those labeling pattern match with the H2B-GFP observation. So DX1 positive slow cycling population are preferentially located in this the LOC region, the region so-called the interscale. The SLC positive population are located in the scale region. So those are fast cycling region. So in those newly identified the marker actually level the slow and fast cycling population respectively. And then we track their fate over 1 year of time, so which corresponds to 20 or 30 times of epidermal turnover to address their long-term stem cell potential. So first we tested DX1 Cure. So DX1 Cure, so this level for a slow cycling population. So after a few days of tamoxifen injection, the tomato positive cell are located in the basal layer, and this cell divide and differentiate upwards to form epidermal clone. And then even after 1 year, epidermal clone are maintained tissue. So meaning that DX1 positive slow cycling cell act as stem cell. Then how about fast cycling population? So in a classical stem progenitor model, fast cycling cell has been considered to act as short-lived progenitor cell. However, what we found here is that SLC positive fast cycling population also act as stem cell. So those cells self-renew and differentiate and then maintain their clone up to 1 year of age, meaning that these cells are also epidermal stem cell. Then, so this is a homeostatic condition, but how about injury? So in the homeostatic condition, DX1 positive slow cycling population and SLC positive fast cycling population, they locate in the different region of the tissue. However, upon skin injury, those cells have a plasticity to compensate each other's function. So when we observe the tissue 1 month after injury, DX1 positive slow cycling populations start to migrate to fast dividing territory and vice versa. And they can compensate their function, each other's function, in response to skin injury. Then interestingly, after 3 months, when skin injury is healed, those stem cell population in atypical territory are not maintained in long term, and they are going back to their original location. So to summarize so far, in a hierarchical stem progenitor cell model, tissue stem cells have been considered to act as slow cycling cell at the top of lineage hierarchy and giving rise to first dividing progenitor cell. In contrast, in the mouse skin epidermis, we propose a new stem cell model. So here, the tissue is maintained by at least 2 distinct stem cell populations that divide at different rate and located in the distinct territory of the skin. During homeostasis, those 2 stem cell populations act independently to each other, but upon skin injury, they also have a plasticity to compensate their function. So after this discovery, we wonder whether this kind of proliferative heterogeneity of tissue stem cell a universal mechanism and apply for other tissue and also human skin. Then if so, what is the biological meaning of having heterogeneous stem cell population in the tissue? To address this question, we first compare our observation in mouse skin and whether this is applicable to human skin. So especially we use mouse tail skin as a preliminary model, and then we wonder whether this is also true for human. So recent single-cell analysis comparing mouse tail skin and human skin stem cell system and their molecular property, there is a molecular similarity and stem cell system similarity between mouse skin and human skin. Then also we identify stem cell markers that are also common in human skin. So here in this picture, we stain slow cycling cell and fast cycling cell marker in human skin, and then found that heterogeneous expression pattern even in the human skin. Then interestingly, in the case of human, those slow and fast cycling stem cell location is correlated with undulating tissue structure. So you can see, so in this picture, the upper layer is epidermis, lower layer is dermis, and the boundary region, there is an undulation. Then slow cycling stem cell population are preferentially located on the top of undulation, and fast cycling population on the bottom undulation. So it seems like even though mouse tail skin and human skin have different anatomical structure, there seems to be a common universal stem cell system across different species. Then we also interested in other epithelial tissue, and that is oral epithelium. So skin and oral epithelium, so both of them derived from the ectoderm origin, and they have a similar the stem cell property. So in the case of oral epithelium, there are also an undulating structure in the tissue, and slow cycling cells are highly enriched on the top of undulation, and fast dividing cell on the bottom undulation. So similar pattern as we saw in the human skin. The another interesting epithelial tissue is a mouse ocular surface epithelium that is corneal and conjunctival. So here we also apply our H2BGFP system, and looking by hormone staining, we found that there is a compartment of slow and fast cycling stem cell in the mouse ocular surface epithelium as well. So together, this result suggests that there is a commonality of stem cell system across different epithelial tissue. Then we are particularly interested in this undulating structure we observe in human skin. And we currently try to mimic this undulating structure in vitro and see whether this micropattern, the environment, is one of the regulators to induce stem cell heterogeneity. So here is a very simple experiment, but very interesting experiment. So here we engineer the scaffold, which has undulating structure. So this we call micropattern gel made with a collagen type 1. And then we coat this scaffold with collagen type 4 as a basement membrane. And then we culture primary human epidermal stem cell on the top of this scaffold. And then we induce differentiation and stratification to make a 3D skin tissue. And then when we observe stem cell property on this micropattern gel. So here's the result. So when we compare with the flat gel, so there are no undulation in this gel. And then the epidermal stem cells still proliferate and differentiate, but are only made the very thin epidermis. In contrast, when we seed epidermal stem cell on micropattern gel, we can see this very beautiful pattern of tissue is formed. So this means that only having this micropattern scaffold, the epidermal stem cells are more activated and then have higher tissue forming ability. Then more interestingly, when we look at the division pattern of epidermal stem cell on this micropattern scaffold, we found that as compared to flat gel, the micropattern gel are able to induce heterogeneous stem cell division pattern. So similar to human skin, so when we culture epidermal stem cell on this micropattern gel, the rapidly proliferating cells are highly enriched on the bottom of undulation, and then slow cycling cell on the top of undulation. So this division pattern heterogeneity is similar to what we observe in human skin. So this observation suggests that those heterogeneous mechanical environment may be among the inducer of stem cell heterogeneity in the tissue. Then next we move on to the aging story. So what is the biological meaning of having heterogeneous population of tissue stem cell? And then we consider aging. So slow cycling or quiescent nature of tissue stem cell are originally considered as an anti-aging or anti-inflammation mechanism, since when stem cells divide more, they might accumulate more DNA damage and mutation, which cause aging or inflammation. So when we consider stem cell aging in skin, the skin actually show very characteristic histological changes during aging. So as I showed you previously, so the young skin tissue have undulating structure as a boundary of epidermis dermis, but this undulation becomes flattened with age. Then, as I showed you before, this undulating structure is correlated with the stem cell location. However, the relationship between stem cell heterogeneity, skin dysfunction, and histological changes of skin remain largely unknown. So we address this very simple question: what happened to epidermal stem cell during the life course of animal? So especially we are particularly interested in whether the slow and fast cycling stem cell undergo different process of aging. To address this question, we perform a very long-term lineage tracing experiment up to 2 years of age. That is the average lifespan of mice. So here, we use the same genetic mouse model, DX1 and SLC1S3 curve, and we label a stem cell by injecting the tamoxifen at young age and then trace their fate over 2 years of age. So first, during homeostasis at young age up to 12 months of age, we found that DX1 positive slow cycling population are located in the interscale region and then maintain their clone within the interscale region. In contrast, SLC1S3 population, they are located in the scale region and then maintain their clone in the scale region. So as I showed you previously, so young age, slow and fast cycling population act to replenish their own compartment during homeostasis. However, when we observe aged mice at the age of 2 years old, we found that this pattern was compromised. So first, we realized in the aged skin, the scale structure, the sizes become smaller, and SLC positive fast cycling stem cell clones are gradually decreased with age. Whereas the DX1 positive slow cycling stem cell clone remain less affected. So this observation suggests that the aging speed of slow and fast cycling stem cell population may differ, and fast dividing stem cells are affected more during aging. In the chronologically aged mice at 2 years of age, the skin also shows delayed wound healing ability as shown by wound healing assay. Then how about in humans? At a young age, the slow and fast dividing stem cell population are located in relations to this undulating structure. However, in the aged skin, this undulation becomes flattened, and SLC positive fast cycling stem cell population are lost. So this is a similar changes as we observed in mouse skin. So as a mechanism of stem cell aging, we found that there is an alteration in stem cell population balance by the loss of fast cycling stem cell population. Then what factor control this process? So our laboratory has been studying the possible regulator of this stem cell fate, identity, and balance of heterogeneous stem cell population. So altogether contributes to the maintenance of long-term skin resilience and their function. So in my today's talk, I will especially focus on the role of ECM. So to address the molecular mechanism of epidermal stem cell, we perform RNA sequencing to compare the young and old epidermal stem cell, and then found that there are many ECM proteins are changed during aging. The amount of ECM, we focus on fibrin 7. So fibrin 7 is a fibrin family of ECM protein and have a conserved fibrin type module on C terminus and coilocoin domain on N terminus. Then fibrin 7 protein is located in the basement membrane, and this expression is decreased with age. To address the functional importance of fibrin 7, we generated fibrin 7 knockout mice and then analyzed their phenotype. So we address the phenotype of fibrin 7 by examining several parameters of skin aging. So first we tested SLC positive fast cycling stem cell clone in fibrin 7 knockout mice, and then found that by knocking out fibrin 7, the SLC positive stem cell clone are prematurely lost at 1 year of age. Then we also tested skin wound healing ability in fibrin 7 knockout. Then fibrin 7 knockout show delayed wound healing, as we show in the chronologically aged mice. At the molecular level, the fibrin 7 knockout epidermal stem cell show the similar molecular changes as we saw in the aged skin, including the inflammatory signatures and the lineage misspecification. Then finally, we address whether the overexpression of fibrin 7 is sufficient to maintain epidermal stem cell in an undifferentiated slow cycling status. So here is an in vitro experiment overexpressing fibrin 7, either full length or fibrin 7 lacking coilocoid domain. Then interestingly, by overexpressing fibrin 7, we found that the epidermal stem cells are maintained in a differentiated state, and the proliferation also becomes lower. So this phenotype of fibrin 7 overexpression is opposite from what we observe in fibrin 7 knockout. And then we identify fibrin 7 binding protein to understand their molecular function of fibrin 7. Then we found that the full length of fibrin 7 interact with other ECM matricial protein, including periostin, CCDC80. Then delta CC fibrin 7. So this is important for maintaining undifferentiated state of epidermal stem cell. The fibrin 7 interact with other basement membrane component, including collagen 4, the tyrosine C, and fibrin 1. Indeed, by knocking out fibrin 7, the basement membrane integrity is impaired, as shown by collagen 4 pattern in this picture. So to summarize the second part of my talk, we have shown that during aging process, the fast cycling stem cells are gradually lost, and there is a impaired stem cell balance happen in aging skin. And this is also true in the case of human. Then we identify extracellular matrix fibrin 7 is one of the regulators of this process. So fibrin 7 is expressed in the basement membrane and essential for maintaining ECM integrity and protects the epidermal stem cell from replication stress and inflammatory environment and prevent the process of stem cell aging. So however, it still remains unknown how those slow and fast cycling stem cell population balance is regulated. So today we talk about fibrin 7 as one of the regulators. Then we also work on the other ECM, fibrin 5 and integrin beta 1. So those factors are very important for balancing the slow and fast cycling stem cell. Then we also identify the metabolic factors as well. One of them is the retinoic acid. So by topical application of retinoic acid also bias the stem cell population towards the fast cycling status. And this is also a strong regulator of those stem cell population balance. During skin development, the balance between Wnt and then EGFR signaling. So those are also very important for the regulation of fast cycling stem cell. And those the signaling is important inducer of fast cycling stem cell, the postnatal stage. So our goal is to identify the target molecule to balance the stem cell population, and also what factors causes the imbalance of the stem cell population in stem cell aging or inflammatory skin diseases. So this is my final slide. So far we use mouse skin as a model system to characterize those slow and fast cycling stem cell population. And then we established genetic tools to visualize those stem cell population and track their fate in different contexts, physiological process and pathological process of aging. And then, interestingly, those slow and fast cycling stem cell population and those tissue structure are dynamically changed during aging. So you can see those aging process by looking at this scale structure. So, and maybe by anti-aging reagent, we may be able to rescue this phenotype, and then those are visually appear in skin. And then we can also combine with the treatment experiment skin and also wound healing assay and aging and disease model. So this is the advantage of using mouse model for study. But we also interested in how to link our basic finding in mice to understand human biology. So that I show you today, so human skin also have a very interesting stem cell system that may be common, some common mechanism between mouse and human skin. Especially in human skin, there is a characteristic undulating structure, and that corresponds to slow and fast cycling stem cell localization. And in case of human, we are able to primarily culture epidermal stem cell and making the 3D skin model, and especially mimicking undulating structure to recapitulate the mechanical environment that we saw in skin tissue. So this is the final slide. So I'd like to thank all my lab members and collaborators and funding support to this project. I'd like to thank all my the audience for this today's symposium. I'll be happy to take any questions. Okay.
Thank you, Dr. Eiko Sada, for your fantastic talk. Now we will proceed to deliver the question from the audience. Le damos el pase a la profesora María Llanos.
Hello again, Dr. Eiko Sada.
Hi. Thank you.
Thank you
Sorry
very
for the
much.
technical trouble.
No problem. Thank you very much for your amazing talk. I'm
Thank
happily
you.
surprised to have the pleasure to see it. I'm excited about your discoveries because it seems that two types of stem cells are responsible for the skin development and going against the injuries.
Yes.
So if you have to think about further studies to disentangle how to regulate these slow growing stem cells, what would be your approach? Because it seems that they are responsible for aging, let's say.
Yeah. So that is a great question. So that is my, life work of my research. So we are currently studying. So let me share my screen again. Can you actually?
Yes.
Yes, great. Okay. So first of all, so we found in aging skin there is a loss of fast cycling stem cell. So our goal is to kind of recover this fast cycling stem cell population even in the aging skin. So that is a basic idea of this our current study. Then how to do this? So we can learn from skin developmental process. So during skin development, those slow and fast cycling stem cell, they are developed from like a homogeneous population, and those heterogeneity develop after birth. And then here, the WNT and EGFR signaling play a role. So for example, so EGFR, signal. So there is a LRIC1. This is an inhibitor of EGFR expressed in the skin dermis, very early postnatal stage. So that inhibits the EGF signal in the slow cycling region. So that is one of the mechanisms to develop the slow and fast cycling population. So the idea is maybe by activating EGFR signal, we may be able to induce more fast cycling stem cell population, and then bias back to this to stem cell population. Then similarly, Wnt signaling is another regulator of those two stem cell population balance. And there is a genetic mouse study overexpressing beta-catenin. So that is a downstream of canonical Wnt. And then those fast cycling stem cell population expand. So if we do this in aging skin, maybe we are able to balance back this fast cycling stem cell. So we think the balance of slow and fast cycling stem cell population are maybe plastic. So if we modulate the proper microenvironment or the including niche signal and the ECM or mechanical signal, maybe we are able to balance back those stem cell population and modulate their identity. Then, of course, there is a transcriptional regulation, and epigenetic factor may also play a role. So we also approach from that point as well. Yeah, thank you very much for your question.
Thanks to you. So how far are we to translate all this knowledge to human skin? I mean, people think about regeneration just not because of injuries, but also because of the beauty and the cosmetics.
Yes, that's also very important issue.
Because as you mentioned, the retinoids.
For us, before I get aged.
Yeah. Okay, me too. So before. As you mentioned, the retinoids. How good are we with these creams or topical treatments?
So retinoid acid. So this is one of the very popular reagents that people use for anti-aging or anti-inflammatory purpose. But retinoic acid signaling in general, if we do experiment in mice, it's actually have an adverse effect as well. for example, if we topically apply electric acids to mouse skin, it's caused a depletion of fast cycling stem cell. So it's not recovering, it's making it worse. So I think this is also dose dependent and also target cell dependent. So it's electric acid signaling. It's a little bit tricky target molecule. Of course, it's activates stem cell proliferation and differentiation. So temporarily activate skin turnover, but in long term, it's deplete the epidermal stem cell population. So it's not super good, I would say. So we might need more alternative, the target molecule that have maybe positive effect on stem cell population, even in the long term. So that's my idea. So maybe we need to test something else. And then the human, if we consider human, one of the difficulty is still there is a gap between in vivo study and in vitro. Then, of course, we cannot do experiment in human directly, so we need some good in vitro skin model. Then in vitro skin model has advanced greatly. However, still there is a big difference between the actual human skin tissue and the in vitro skin model. So if we compare, for example, epidermal stem cell population in vitro skin model, so still there is a big gap. I think it's because the environment is not fully recapitulated in vitro model. So we need maybe more factors to really model those human skin in culture dishes. So that is a current challenge. So once we have those system, we can test those, target molecule, identify in mice, and then translate in humans. So those bridges maybe become more easier.
Okay, we have one more question from
Yes.
the audience. Okay, so let me check it out.
Yes.
Okay, let's see. Wait a second. Oh, what implications could these regulatory mechanisms have in tissue regeneration or in disease?
Disease, yeah. So we are also working on inflammatory skin diseases as well. So we collaborate with skin dermatologists and then analyzing human patient sample. So this is highly actually disease dependent. So some skin diseases like psoriasis, the slow and fast cycling stem cell population balance also change. And then we are currently demonstrate that this fibrillin 7 molecule also have an anti-inflammatory effect, not only anti-aging. So some of the molecule we identify in aging study maybe also have an effect in the skin inflammatory disease as well. So we are currently working on several skin diseases, but the stem cell changes seems to be different between different diseases. So we need to characterize probably one by one. And then different immune cells also play a role, and then different cytokines play a role. So it's a little bit more complicated. But I think we need to figure out what happened in human patient case, and then whether we could model in our mouse studies. Yeah. Maybe next 10 years.
Or specifically for, okay, it's incredible because it has to be like specifically in each disease, how the regeneration
I think
happens,
so.
right?
I think so. Yeah, seems like regulation is a bit complicated, and it's really context dependent because the different immune system play a role, and the different immune cells and cytokines play a role in different skin diseases. So phenotype and stem cell effect seems to be different. Yeah.
Okay, so I have another question, a last question, I guess. Let me check. So how could modulation of these pathways be used for regenerative therapies?
Sorry, could you repeat that question?
Okay, sure. Please. Okay, so how could modulation of these pathways be used for regenerative therapies?
So regenerative therapies, that's something I'm not actively working on. So regenerative therapy. So there are several approaches. So one is transplantation. So making skin model, like our 3D skin model, and making 3D skin and transplant back to patient. So that is one of the most common regenerative therapy in skin. And then the another approach is to target molecule like we do. So we identify the stem cell regulator, and then we kind of recover those stem cell potential and recovering the stem cell regenerative ability, so let's say. So this is kind of similar idea as we do in aging study or inflammatory disease study. So, but again, it's a context dependent. So skin injury. So it depends on how deep, skin injury go. If it's an epidermal only injury, or it's a full thickness wound, or diabetic wounds is a very scarring wound that's very hard to heal. So again, it's kind of context dependent, and then stem cell behavior actually very different in different wound models. So I think in each wound model, we have to identify the factors, and then that factor may be a therapeutic target for those skin degeneration part as well. So we are currently working on that.
Well, it seems that's a lot of work to do.
Yeah, a lot of work to do.
It's so exciting.
20 years.
Yeah. Okay, Dr. Aiko-Sada, it was a great pleasure talking to you, and I would like to say thank you in behalf of the chair of the Cellular and Molecular Biology from our university, the National University of Trujillo. It was a pleasure. It was an honor to have you. And I hope it's not the last time.
Yeah.
But we have many other opportunities to talk to you, to exchange experiences. Thank you so much. Deeply.
Thank you very much for inviting me to this wonderful symposium. I'm very glad to see you all. Okay. Thank you very much.
Thanks to you. Till next time.
Thank you. Enjoy the rest of symposium. See you.
Bye. See you.
Bye.
Thanks.
Buenos días a todos los presentes. Lamentamos informar que debido a motivos urgentes, la doctora Irene Julka no podrá presentarse durante esta oportunidad. Ofrecemos nuestras más sinceras disculpas por cualquier inconveniente que esto pueda causar y agradecemos su comprensión. Esperamos contar con su presencia para nuestra próxima charla que corresponde al doctor Ricardo de Liponti de 10 a 11 de la mañana. Muchas gracias por su atención.
Buenos días, bienvenidos nuevamente al quinto Simposio Internacional de Biología Celular y Molecular. Esta conferencia será realizada en inglés y es así que desde este momento hablaremos principalmente en este idioma y por ello se les recomienda activar la opción de subtítulos. Good morning. It is a great pleasure to introduce Dr. Riccardo Deli Ponti. Riccardo Deli Ponti graduated from the Università di Roma Tor Vergata with a bachelor's degree in biotechnology in 2009. He obtained his master's degree in bioinformatics in 2012 and his PhD in biomedicine in 2018. Currently, he is working on RNA design, vaccine optimization, and feature extraction of potential therapeutic target regions. Dr. Deli will deliver his presentation titled Predicting the RNA Secondary Structure of RNA Viruses. Dr. Deli, we warmly welcome you to this event and sincerely thank you for your participation and support. We would like to remind you that you have a maximum of 45 minutes for your presentation, followed by a 10-minute Q&A session. To the attendees, kindly ensure that your microphones and cameras remain off to avoid interruptions during the presentation. Please submit your questions via the chat throughout the session. They will be addressed at the end of the talk. Additionally, a link to the attendance form will be shared during the presentation for you to register your participation. Al público presente en la sala, les recordamos mantener sus micrófonos y cámaras apagadas para evitar interrupciones durante la exposición. Si tuvieran preguntas, estas deberán ser formuladas mediante el chat durante el transcurso de la presentación y serán absueltas al finalizar la ponencia. Además, les comunicamos que durante el transcurso de la exposición se les estará compartiendo el enlace al formulario para que registren su asistencia. Dr. Deli, you may begin with your presentation. We are eager to hear from you.
Okay. Thank you for the introduction. Hi, everybody. So, well, I will switch now on a second, but then for better connection, I will switch off the camera. Okay, I will start to share my slides. Let me know if you can see them. 1 second. Can you see my screen?
Yes, we can see
okay,
the screen.
Perfect. Okay, so again, thank you for the introduction and for inviting me. So today I will speak about some of the projects that I worked on in the past year, and I will try to show you algorithms to predict the RNA secondary structure and how we apply this algorithm to predict and to characterize RNA virus. So just to show you some images about where I worked, about my places, let's say. So I'm originally from Rome. I did my PhD in Barcelona, so right here at the CRG, and then I moved for an internship in Japan at RIKEN, and then for 4 years in Singapore. You can see here, this is an image of the Nanyang Technological University, but not in a building that I worked on, but yeah, it's a very emblematic one. And now I'm working at the IIT, so the Italian Institute of Technology in Genoa, which is built on top of a mountain. So today I divided the presentation into main topic. So in the beginning, I will speak about the algorithms that I developed during this year. Some have AI or machine learning, it depends which word you prefer. Others are more derivative tools, so based on a combination of previous tool or workflows or pipelines. And then the second part of the presentation, it will be more of a computational analysis, so how we use these instruments to characterize, to work on RNA viruses. So all the work that I will show you today, it's completely computational. So I will try to give you some ideas of I don't know how much you are familiar with it. I will try to give you some insights, but I also will try to give a general overview. So some things I will just give some details. So if you are interested, you can then later ask more about it. So these are the algorithms that I developed. So as you can see, 3 of them are called all CROSS. So this is because it's a suite of tools. So CROSS is a suite of tools based on predicting different structural features of the RNA. while RNA Navigator, it's more pipeline, so based on existing tool. So why we want to predict their secondary structure? Why is this useful? Well, the RNA, this RNA structure is far more complex, of course, compared to the DNA one, and it has more degree of freedoms and can takes many more forms and shapes. But knowing the secondary structures allows us to understand a lot of more feature of the RNA. For example, we can know the 3D structure of RNA by knowing the secondary structure. We can already infer the interaction of RNA with the protein partners or the interaction between two different RNAs. Also, the secondary structure is very important for the to study evolution and conservation, because a lot of time sequence conservation is not enough. Let's just think to viruses, for example. So they mutate a lot, and the structure sometimes it tend to mutate less. So it's important also to know for an evolutionary point of view, and as well for other more peculiar features such as scaffolding and the formation of stress granules, for example, are also based on very structured and long RNAs. So for all this reason, it's very important to know and to understand their secondary structure. But not always we have time or access to experiments, so it's also important to predict the RNA secondary structure. So this is why I developed CROSS. So CROSS, it's a neural network to predict the RNA secondary structure. Of course, it's a project which started in 2015. So if you are familiar with all this AI and deep learning, this is like in a time when it was not that fancy, but still it's based on neural network approach. The idea of CROSS is to predict, the output of CROSS, it's a profile, and for each nucleotide, you have the propensity of that nucleotide to be in a double or single-stranded conformation. Since the output is a profile, it's a very fast tool. For example, it can profile exist, which is a very long RNA, around 18,000 nucleotides in a couple of minutes. But the very novelty of CROSS is that it was trained on 5 different experimental data sets coming from different techniques, for example, shape, parts, NMR, X-ray techniques, and also different organisms. For example, we have parts both applied on yeast and human. And while we have these 5 different data sets, we also have a global score, so an ensemble approach which combines the 5 different predictors into an overall score. Very few details about how CROSS was built. So the idea behind CROSS was to select all these structural profiles from different experiments and identify the structural peaks, so nucleotides which were in a high propensity to be in single or double-stranded conformation according to the different technique. Around that nucleotide, we selected a fragment. So the input it's of CROSS, it's just the RNA sequence. So this fragment is then converted using a one-hot encoding procedure. So each nucleotide is converted in an array of 1 and 0s. And this is the input of a neural network. So a fully connected one direction neural network. And the output is the propensity of that specific nucleotide at the middle of the fragment to be in a double or single-stranded conformation. So after developing CROSS, we started to test it, to apply it on different and complex RNAs, so viruses, RNA viruses, and long-encoding RNAs. Here you can see in the top picture the HIV-1, so a section of HIV-1, and you can see the predicted profile and the experimental profile. So they have a very high correlation, meaning that CROSS is able to predict the structure of HIV. Here you can see instead exist, so a very long RNA, long-encoding RNA. And as you can see as well, here we have the performances on another technique called DMS, which was not present in the training set. And we have an AUC. AUC means area under the ROC curve. So it's a metric often used to establish the power of a predicting algorithm. So the area under this curve is 0.75. So meaning that we were able to reproduce, to predict also different experimental data. And here you can see a typical CROSS profile. But also cross was very interesting because here you can see an RNA, the real structure of RNA in a circle plot representation. And while cross was the first tool to be trained on experimental data, several tools based on thermodynamics were mainly used to predict the RNA secondary structure. So they were not based on experimental data, but more on thermodynamics laws. Here you can see the structure predicted by one of these tools called RNA structure. So while, for example, this domain is very well predicted, the other domains, the orange one, are very disrupted. But interestingly, you can feed to this thermodynamic-based algorithms experimental data. So if you have your experiment, you can feed then the data to this algorithm, and they have any better predictions. So we did the same with cross. So we try to simulate, in this case, were shape data, so data to predict the RNA secondary structure, using our cross output, and then feeding this output inside the RNA structure. As you can see, there is an improvement in the prediction. Of course, here you can see a more overall picture. So, for example, if we use as an input inside the RNA structure the experimental data, so shape data, of course, we have a huge improvement, but these are experimental data, so come with time and money, and of course, all the golden standard. But if we use cross, we also have an improvement compared to RNA structure alone, meaning that cross can be also used as an in silico alternative to shape data. So this was a very interesting output, and also something that you can use like to improve other softwares. Even if cross was developed some years ago, it was very well used by several collaborators. For example, it was used to assess the RNA secondary structure of Samson, which is a long non-coding RNA associated to cancer. Other long non-coding RNA also were profiled. For example, this one responsible of Italian differentiation, and even very recently, it was used to study the composition of stress granules, so how the RNAs inside the stress granules are from a structural point of view. So the second algorithm I will talk about is cross-align. So this one, it's not a machine learning based algorithm, but it's like a derivative tool. So in this case, it's a combination of cross and a dynamic time warping algorithm. And the idea is to build an algorithm to then compare RNAs of different length, of course, from a structural point of view. So you have two different RNAs, you want to know how similar they are in terms of structure, and you can use cross-align. So here you can see some of the output of cross-align. So for example, here you have two RNAs, in this case are similar length. And here you can see, of course, the more this path is close to the diagonal, the more similar they are. Here you can see the region which is more similar, it's very well represented. And also it has a function to look for structural domains. So if you have an RNA, cross-align will fragment this RNA in different subregions, and then we'll search each of these regions inside a bigger data set, for example, all the human long non-coding RNAs, to see which regions are more, let's say, conserved from a structural point of view. So after we developed cross-align, I mean, the first test was to study known domains. In this case, we select the RepA. So the RepA is a domain of XIS as well. You probably are very familiar now with this name. So we searched the RepA of XIS, but the RepA of orangutan and the RepA of baboon. And then we searched this RepA against all the human long non-coding RNAs. And as you can see, XIS is found as the best. Human XIS always has a best result, meaning that our algorithm is able to recognize this structural similarity. The last test we did of cross-align was on viruses, in this case, again, HIV. So what we did was to compare around 4,000 different strains of HIV. And here you can see how they are similar in terms of sequence and in terms of structure on the x-axis. And the interesting detail is that you can see there are some clusters, for example, here around 0.09, These strains, they are more similar in terms of structure than in terms of sequence, while these other two here, they tend to be more similar in sequence. The sequence is changing less than the structure. This is highlighting how sometimes sequence and structure are not bringing the same level of information, meaning that not always sequence and structure follow the same conservation pattern. It is very important to know the conservation, to study conservation both of sequence and structure, especially on viruses. The last tool of the Cross suite is CrossAlive. Again, we are talking about machine learning approach in this case. So this is an AI. And the idea of this tool was to study the RNA secondary structure to predict or to better predict the RNA secondary structure, but in vivo, so using in vivo data. Of course, this RNA structure is very different from in vitro to in vivo because, of course, the cellular environment is very different from temperature. Of course, it's a more crowded environment, so there is the presence of ligands and proteins. So, of course, the structure is also affected by all this very different environment. And it's even, you can see it also from a computational point of view, because here you can see all these arrows are testing of methodologies against this other, and these are the accuracies of different models. But the message is like, when we try to predict in vitro data, it's easier, so we have better performance than when we predict in vivo data. In this case, the data we use are IC-shaped data, which were provided both with and without methylation. So this explains why we have plus M6A and minus M6A. But the message is like it's more difficult to predict in vivo data, and you can see even better here, compared to in vitro data. This could be because, of course, the environment is very different, and maybe we are missing pieces of the puzzle. So the idea we had at the time was that one of the pieces of the puzzle that we were missing was the presence of the proteins. So we wanted to build a new cross that not only used the information coming from the RNA, in this case the RNA sequence, but also the information coming from the proteins interacting with it, which could explain how the structure in vivo was more complex to predict. So what we did, on one side, we have a similar model to Cross one. So we have a profile, and we have the peaks of this profile. We select a window of nucleotides around these peaks, which were converted in one-hot encoding in 1 and 0, and this is part of the input. But on the other side, we have the information coming from the protein. So for all these fragments, we predicted the binding with a selected range of human proteins, which we already know were able to discriminate between RNA structures. And here you can see the values of the predicted interaction. And this also is part of the input of the network. So on one side, we had information coming from the RNA. On the other side, we have the interacting proteins. All this is the information inside the network. And this is what the base of CrossAlign. So here you can see the new output. So when we in this case, when we have the information coming from the proteins, when we add the information coming from the proteins, we have better predictions compared to not using protein, both with and without methylation, M6A. Again, we have here our favorite exist. These are in vivo data, DMS in vivo data, if I'm not wrong. As you can see, we have a very high AUC, and this is the profile. So CrossAlive is a very interesting method, able to predict the RNA structure in vivo, combining information coming from the RNA sequence and the interacting proteins. We also did an update last year, so about CrossAlive. So since we have at the beginning data with and without methylation, we also decided to include data with and without another modification, in this case, A2I modification. As you can see here, if we study A2I, especially for long non-coding RNAs, we see that when the A2I modification is present, we have an increase in the structural content, meaning that there is an increase in structure in double-stranded nucleotide when the modification is present, meaning that this modification could promote the formation of secondary structure. And this is evident even if we select specific non-coding RNAs, which are very studied, like NORA, NIT, and our favorite exist. And yeah, this was a model, a new model, so based on the cross-alive architecture, which was released last year. The last tool I will talk about, it's RNA Navigator. So RNA Navigator is not part of the cross suite, but still it's using information coming from the secondary structure. The idea, this is again, like cross-align, this is a derivative tool, so it's more a pipeline or a workflow, and the idea of the algorithm, it's like to identify potential regions, functional regions on a novel RNA. So imagine you have a novel RNA, like a novelly discovered non-coding RNA or a new virus, and you want to know if there is a region that could be of interest on its genome or RNA sequence. You can use this algorithm. So this algorithm takes 3 different layers of information. So on one side, we have the information coming from the secondary structure. Then we have the structural stability predicted with scanfold, and then we have the entropy predicted with superfold. And the idea, the interesting idea behind this tool, it's also that it can run with and without experimental data. So if you are a lab which can perform experiment, shape experiment, you can feed your own experiment inside RNA Navigator to predict regions of interest. Or if this is not possible, it runs cross to simulate shape data. So you can have the predictions. So let's see a typical output of RNA Navigator. So here we have dengue one, so the dengue virus, so flavivirus RNA. And here you can see the 3 different metrics, so entropy, scanfold, and secondary structure, and the regions of interest for the 3 different metrics. Here we have with experimental data, shape data, and here we have the prediction with cross. And it's interesting to see that the 3' UTR, which is a regions that it's known to have a lot of secondary structure which are functional for dengue viral infection, are very well predicted. We also did this on other viruses like hepatitis C and non-coding RNAs, but I will not show you all this. So this concludes the first part of my talk. So I show you all the algorithms that were developed to predict, compare, and characterize the RNA structure, the RNA secondary structure. Now I will talk more about analysis that we did using these instruments on viruses, on RNA viruses. So I will talk mainly, but not only, about flaviviruses. So which are flaviviruses? So flavivirus is a class of RNA viruses, single-stranded and positive-stranded RNA viruses. They are usually arbovirus, so this means they are transmitted by a mosquito bite. The most famous one, according to CDC, it's yellow fever, dengue, Japanese encephalitis, West Nile virus, and Zika. You probably know some of them. And their genome is quite simple, so they have a 5' and 3' UTR, and then the coding region which encode for structural and non-structural proteins. But I will focus more on the genome more than on the proteins because I work more with RNA. So the first work that I will show you, it's about the characterization of RNA viruses, so mainly flavivirus, but not only. So we're trying to characterize, let's say, viral hemorrhagic fevers, of course, of different intensities, including Ebola virus. The idea behind this project was to move away from the sequence information, let's say, and to try to characterize RNA viruses from a structural point of view, and also using information coming from the interaction with the human proteins. So we wanted to find new metrics to clusterize and characterize and compare RNA viruses. So here you can see the predicted structural content. So here we have around 8,000 different genomes from 11 different species. And you can see some of them, like the 4 dengue serotypes and Ebola, they are very unstructured, so the structural content is lower, for example, compared to Japanese encephalitis or tick-borne encephalitis, meaning that there is a different level of structural information for each virus. And here you can see how the structural content also compare with the number of protein binding motifs. So we checked the presence of known binding motifs on the RNA genome of these viruses, and we average, of course, the presence of motifs compared to the average genome of each virus. And here you can see in the 4 dengue serotype how each serotype has a very specific, well-defined cluster where we compare structural content and the number of protein-binding motifs, and also with a different correlation between them. So this is very interesting. Between the same serotype, it means that there is a different level of characterization. And when we use this information to cluster viruses, using the presence of protein-binding motifs and the structural content, as you can see in some cases like dengue 1, 3, and 4, the comparison is very similar, so they form a similar cluster. But for example, dengue 2 is more similar to chikungunya for the presence of binding motifs, for the structural content, it's similar to Ebola virus. And this is quite interesting because dengue 2 is the most aggressive, the most prone to give hemorrhagic fever of the 4 dengue serotypes. So it was a very curious result. And to conclude about this, I can show you again the structural content and the presence of protein binding motifs for all the different viruses, and you can see there are very different trends. You can see especially chikungunya and Zika, they are the two opposites. So for chikungunya, the more unstructured the genome is, the more it's prone to bind more proteins, so there is very high anti-correlation. And on the other side, the Zika, instead, the more structured the genome is, the more prone it's to bind more proteins. Meaning that there is a very peculiar signature that is different between the different viruses. So I will briefly talk now about SARS-CoV-2 for its importance. So this is a project which born at the beginning of the pandemic, so 2020. So of course, when SARS-CoV-2 appeared, we tried to use all the instruments that we had at the time to characterize it and to help then further experiments. So while of course the world was waiting for more and more experiments, so what we did here was to predict the secondary structure both in vitro and in vivo of SARS-CoV-2. And also we compare SARS-CoV-2 with all the known coronaviruses. So here we use cross-align, so we divided the genome of SARS-CoV-2 in all these subregions, and then we search each of these regions, how they were conserved in all the, of course, from a structural point of view, in all the known coronaviruses. And you can see the regions encoding, for the spike protein, which then was one of the most, of course, important regions for vaccine development, was also the most conserved in terms of structure. And yes, this concludes the part about SARS-CoV-2. So this is the last project I will talk about today. So I think I'm very okay with the time. So this last project is about, again, flaviviruses, but about a specific mechanism of flaviviruses. So the flaviviruses, where they are in the cell, the genome have this very peculiar mechanism. So you can see XRN1, so the human protein, it binds to the genome and it starts to digest it, so while it's in the cell. But then it happened, and there is a very peculiar set of structure at the 3' UTR of the flaviviruses, and then this structure stole XRN1. And so this fragment called flaviviral subgenomic RNAs, they start to accumulate inside the cell. And it was shown in different publications that these subgenomic flaviviral RNA are essential for the anti-host defense response of flaviviruses. For example, in West Nile virus, if these subgenomic RNAs were degraded, it lowered a lot the virulence of West Nile virus. And also it was shown in different publications that, for example, in dengue and Zika, using only one genome, it was shown that these subgenomic RNAs were prone to bind with human proteins and to disrupt some biological processes by doing that. So what we wanted to do for this project was to study more this subgenomic flaviviral RNA, so this fragment at the 3' UTR of flaviviruses, and to study, but at a high throughput level, so using more than 8,000 genomes and more than 2,000 human proteins, to study in silico how these fragments bind human proteins, and if this has an anti-host defense function, and at which level this binding can be considered as a sponge for human proteins, so how prone are to bind human proteins. So here you can see on the right, this is all the interaction between dengue subgenomic flaviviral RNA and the human proteins. Interaction propensity, of course, the higher is the score, the stronger is the binding. Here you can see TRIM25, which is a known experimental validated binder of subgenomic RNAs of dengue. It's very well predicted, so it's a very high interaction propensity in our set. And so we also tested we did a lot more tests, but I will skip it. So then what we did was to select, sorry, I forgot to say that the flaviviruses that we use in this analysis were the 5 most known according to CDC, or most studied according to CDC, so yellow fever, Zika, dengue, Japanese encephalitis, and West Nile virus. So for each of them, we selected the protein with the highest score, so the one with an interaction, with a zeta score, so an interaction propensity higher than 1.5 in our dataset, and we selected and studied all the possible combination of proteins. So for example, we have some proteins like this 97 here at the end, which are specific only on West Nile virus. So this 97 protein, they bind very strongly and very specific only West Nile virus. While we found 5 proteins which are in common between the 5 flaviviruses, meaning that it's a very broad interaction. And here you can see the 5 proteins. So we have especially helicases, and also we have some proteins that are known, several of them are known to be part of the host defense response. So means that it's the binding with this protein can already be disruptive for the host. But then what we did was to focus on 4 only flaviviruses. Why? Well, just because yellow fever seems to be very poorly interacting with human proteins compared to these other 4. So to enhance the pool of proteins, we selected only the 21 which are highly interacting between these 4 flaviviruses. We characterize this protein very well. So here you can see the geo they are associated. So, and here you can see more detail, but these proteins are all associated to very important, crucial mechanism, cellular mechanisms, so that are crucial for the host cell, in this case human. So of course, if they bind to this subgenomic RNA, means they cannot do their function, and this of course is causing problem to the cell, to the human cell, meaning that the flaviviral RNAs, subgenomic RNAs, tend to have anti-host defense function. And even more interestingly, if we take these 21 proteins that we found and we use them as a tool, so imagine as a predictive algorithm, they have indeed a function, so they are a signature. So I will explain better. If we take another flavivirus, not from Southeast Asia, not from South America, but the Murray Valley virus, which is typical from Australia and was not present in the previous set that we analyzed, we take this 25 protein, we run prediction against this Murray Valley virus, and we see where these proteins are highly interacting. We found that in the 3' UTR, the regions where these subgenomic RNAs form, they are all of them are highly interacting. And it's very interesting because cannot be explained because the sequence similarity of flavivirus is around 40%, so cannot be explained by sequence similarity alone. And it's even more interesting because if we do the same thing, but with another virus, so not a flavivirus, in this case HIV-1, I found none of these proteins are interacting with HIV genomes, meaning that these 21 proteins can be a signature identifying for subgenomic RNA forming regions, meaning that in future, the different set of proteins interacting with viruses can lead to identifying a signature for a specific function. The next step we did about flaviviral subgenomic RNAs was to check how strong is the binding with the human proteins. So we wanted to see, okay, they bind with human proteins, with a lot of human proteins, but is this really a lot? How can we say this is a lot? So we needed an index. So we built this binding promiscuity index, which is the number of strong interactions, so a zeta score higher than 1.5 divided by the number of sequences that we have in our dataset. And here you can see plotted here this BPI, so binding promiscuity index for the flaviviruses, and then for some controls. For example, which is the first control. Here you can see there are the coding RNAs, so the last, the 3' UTR, let's say, of coding RNAs, but only selecting the coding RNAs that, according to experiment, in this case, ECLIP experiment, are the most interacting, so the 100 most interacting RNAs. So, of course, they are the top one in this analysis, but this is what we expect because we are selecting by experiment the one that are mostly the highest interacting with human proteins. And after that, we have West Nile virus. So this is also very interesting because West Nile virus, it interacts more with human proteins than coding RNA of the same length and long non-coding RNAs of the same length, meaning that it's really acting as a sponge for human proteins. So why West Nile virus? It's interacting so much with human protein, and what this could mean. So what we did as a very last analysis was to study the propensity of these 97 proteins that were highly interacting only with West Nile virus subgenomic RNAs, if they are prone to phase separate. What means if they are prone to be inside a stress granule. Here you can see with cat granule, which is a tool developed in the lab, not by me, you can see all the proteins, how they are predicted to form, to phase separate, let's say to form granules. And you can see the majority of these 97 proteins have a positive score, so they are prone to form granules. And we also ran a test, so we did random sampling on 97 proteins in all the human protein, and we checked each extraction, how many of them were with a score higher than 0, and in the majority of time they are a lot less than the one in West Nile virus, which is this dashed line. What this means, this is just a statistical test to say, okay, this is significant, so this result is significant. And what does this mean? Well, it is known from literature that West Nile virus doesn't promote the formation of stress granules. So after an infection with West Nile virus, the stress granules are not forming. And the stress granules are can be considered also a defensive mechanism of the cell. And the fact that the subgenomic RNAs are binding a lot of proteins which can be prone to form granules means that subgenomic RNAs could also in part explain why West Nile virus it's not doesn't promote the formation of stress granules. And yeah, I think that's it. So yeah, I finished a bit before, but well, if you want more time for question or more time for you to rest after. So this is a picture, one of the most recent of the group, where we were having the retreat in Slovenia. And well, and this is another picture, one of the historic one, which I found pretty funny. So I would like also to conclude with this one. And yeah, I will stop to share if I found again the page. And yeah, that's it. Thank you for the attention.
Thank you very much for your presentation, Dr. Deli. We will now proceed to address the audience's questions. Le damos pase a la doctora Llanos.
Good morning, Dr. Deli Ponti. It was a pleasure to have your talk. Now I'm going to translate the questions that the audience has. So the first question that we have is the following. Let me check it. Okay. Is there any common feature in the secondary structure that RNA viruses share? If so, what evolutionary significance would it have?
Yeah, as I said before, viruses tend to evolve a lot, so it's very typical between the different viruses. What we can say is that it's often there are structural information inside the UTR. So 5' UTR and 3' UTR are usually the regions which are known to host for very specific structure, which can or cannot be conserved. For example, in the 5' UTR, we have a lot of time the IRS sequences, so which are necessary for the translation of viruses. They tend to be in the 5' UTR, and they can be conserved to a different extent between different viruses. Then, of course, the structure that I told you about, so this XRN1 stalling region, it tends to be conserved to a different extent, but some specific part of it are conserved among flaviviruses, but are not found in other viruses, for example. So it all depends on the viruses. Also, they evolve a lot, but the general view about evolutionary point of view is that while the sequence can evolve a lot in viruses, sometimes the structure, or specific part of the structure, they tend to be conserved. So it can be interesting to study more the evolution of viruses from a structural point of view sometimes.
Okay, thank you. So I have another question, maybe for someone who is interested in starting this approach that you develop in the lab, right? What was the most challenging part when predicting the secondary structure in vivo and in vitro of these viruses?
Okay. Yeah, a lot of times. So now, of course, a lot of methodology evolved. So there are a lot more instruments like deep learning. There is really a word behind the instrument. But what is always complicated, it's at the very beginning to balance, let's say, the data. So the data that you want to use to predict something with then the architecture that you want to build. For example, in my case, I had a lot of data, and what was very useful then to make the prediction works though was to filter out all the noise. So you have like, I don't know, millions of data coming from all the organisms, but then the majority of it was like noise, so it was not helping to do prediction. So a very important step was to filter out all the noise and then to come up with, instead to have millions, to have like, I don't know, 20K of data points and use that to predict. Right now, you can even do, if you have a very power, I mean, if you are Google, you just take all the internet and you train and you don't mind the noise and something will come out. So it depends also on the power of the architecture that you want to use. But yeah, in my case was the filtering.
Okay. Well, thanks for your work. Then it's easier for the future generations to do this kind of stuff. By the way. Yes.
Well, yeah. Okay. Thank you.
Yeah. So I have another question.
Yeah, sure.
What are the criteria to take into consideration when choosing a tool for doing this kind of predictions?
I would say more it's not that, I would not say even depends on what you want to do, because a lot of, yeah, it a bit depends on what you want to do. So there are some architecture right now that, for example, they work better with images like convolutional neural networks, some that work better with like data that are, time constrained data. So they are based with the information coming before. So it depends a bit on what you want to predict. But what I want to say more, I think it's like, if you want to build something, you need to read the papers about it. So a lot of time you find the information you want, maybe in a paper on a different topic, but maybe they use an architecture. Say, oh, well, this can be also working for my data. Just I have to arrange it a bit. So a lot of time, I think, reading about the different methodologies that are used, the golden standard, even in different fields, like, I don't know, physics or chemistry. It can be very useful also then to apply to your data. So I think reading it's very important then to decide what instrument to use.
Okay, thank you very much. So in behalf of the organizers, I am part of the committee. We would like to thank you a lot for your time, your effort to present and stay with us today and share all your knowledge. I hope in the future we have the chance to meet you again, to have another talk, and it would be fantastic. Thank you so much. Till next time.
Thank you so much
Bye.
for the time. And
Bye.
bye-bye.
Okay, we extend our heartfelt gratitude to Dr. Deli Ponti for his excellent presentation, which I am sure has been enriching for all of us. Thank you for sharing your time, knowledge, and expertise in the field of cellular and molecular biology. We also thank all attendees for joining us this morning. We invite you to stay with us for the next presentation. Agradecemos también a los participantes de esta mañana por su asistencia. Invitamos a todos los presentes a continuar acompañándonos con la presentación de nuestro siguiente expositor.
Buenos días a todos los presentes. Lamentamos informar que debido a motivos urgentes, la doctora Verónica Eisner no podrá presentarse como estaba previsto. Ofrecemos nuestras más sinceras disculpas por cualquier inconveniente que esto pueda causar y agradecemos por su comprensión. Por ende, hemos concluido con las ponencias de esta primera sesión de manera muy satisfactoria en el quinto Simposio Internacional de Biología Celular y Molecular 2024, con la gratificación que produce el deber de difundir el conocimiento científico y de crear un espacio que fomente la discusión de temas actuales de gran relevancia dentro del campo molecular y celular. Les agradecemos a las personas que se encuentran con nosotros presentes en las diferentes plataformas de transmisión por la atención brindada. Y de manera muy especial expresamos nuestro agradecimiento a los ponentes, quienes tienen nuestro reconocimiento y agradecimiento por su apoyo en la divulgación del conocimiento científico de manera gratuita, el cual garantizó el éxito de esta jornada. Asimismo, felicitar a los intérpretes por su extraordinario trabajo y su espíritu de colaboración. Finalmente, les invitamos a seguir acompañándonos hoy a las 6 de la tarde, hora en Perú, para poder continuar con el desarrollo de este evento. Muchas gracias a todos. Buenas tardes.